Good morning, everyone. Welcome to Electron Microscopy and my tribute to the Beatles, so here, there, and everywhere. I hope the pun works. Do we have someone from PixelGen here? They are the sponsor. Okay. So if you want to come up, and I'm just going to do some housekeeping, rules and regulations while we get going. The plan is we're going to have the sponsor talk for 10 minutes, and there's a timer running on the screen down here, and then we have 3 presenters, each of whom are going to have 20 minutes to present. And the idea of the session is that we have a breakout this afternoon. So the plan is to continue the discussion and come up with new thoughts and ideas at the session this afternoon. So we're going to fill this morning with the talks. So first, let's just quickly go through some ABRF housekeeping. Phones, please keep everything on silent so we get no interruptions. You are allowed to take photos and video of the presenters and their talks unless they say otherwise. Social media policy is that everything can be shared. Again, just take people's personal preference into account. Questions, we might have a few minutes for a couple of questions at the end today, but don't particularly plan on it, but we will not be having questions at the end of each presentation. You can download the ABRF app to get the full program, find out what rooms everybody's in, and find our session for this afternoon. And please rate the presentations in the app. Those are used by ABRF to think about future topics and future sessions at next year's meeting. Okay, so let's move along. And our first presentation, well, I'll let you introduce yourself.
Hi, everybody. I'm Steve Glavis. I'm actually coming from Sweden these days. But I work at PixelGen. It's a new company, a startup, founded in Sweden just recently. Let's see, how do we get this thing going? Where's the, do you know which button you press?
That's a good question.
The big green button is not working. There we go. Let's go back a little. All right, so we were founded in 2020, about 25 employees or so, and we're based at the Karolinska. So the whole process, what we're trying to do is, what we want to do is start changing things that you're doing with the central dogma. What we want to focus on is more the phenotype of cells rather than the actual genotype or RNA expression or any just standard protein measurements. But some concepts actually put into place, something to think about, is that what we want to focus on is the fluid bilayer, so it's all in motion. And what we do is we lock it in place with PFA, so we fix it. And then what we want to do is take a snapshot of those proteins and then read them out with our next generation sequencer. And things that we can measure are abundance, the spatial distribution, as well as the co-localization of the different proteins on the cell surface. So abundance you can use a flow cytometer to do, and for spatial distribution and co-localization you can use microscopy for. But we like to use next generation sequencer to do this. Some concepts that we bring in are polarization as well as co-localization. And polarization is a term that we use for patchiness, so protein rafts or things like that where they collect on one side of a cell rather than being evenly dispersed. And the higher the number, the more polarized the cell would be. We also have a term for co-localization where you have proteins that group in pair or three-way, so it really depends on how you want to analyze this. And that's useful if you're doing immunotherapy where you're building antibodies that are bi or tri-specific. We like to say that our particular solution fits in the this part of the market. So you have site-seq and other high plexi type of analyses for single-cell applications. It's on the high end. We also have other systems which are microscopy-based. They actually take images. We're able to do both in the same assay. But the assay is based off of a technique called molecular pixelation, and that's a new molecular technique which you can find the details in this preprint on bioRxiv. I won't go into details here, except describe the basic workflow, which is we fix the cells with PFA, we stain the cells with antibodies with oligo tags associated with them, and then we have a special reaction called molecular pixelation where we introduce barcodes that propagate across the cell surface to allow us to generate a next generation sequencing library, and then we read it out with an Illumina sequencer. What we end up doing is getting an edge list, and we use the components to build a graph view, a representation, a relative representation of where those proteins are and what they're co-located with or polarized with. And as a result, we can measure the abundance as well. But the output requires about 1,000 cell output, and we also need around 120,000 cells, 120,000 reads per cell. So you can say a typical reaction will require about 120 million reads. And it's supported on any standard Illumina sequencer, with the smallest being the NextSeq 2000. But if you're a little wary about doing the analysis, we've built a nice pipeline. I like to say it's got a magic green button that you press, and it solves everything for you. But if you think that's not true, there's tutorials online that can help you out, get you through that. And we also have all the data sets available as well. Some of the applications we can work with are single cell level resolution, so it's, we don't go into too deep on the single cells in this particular example, but our pixels can actually give you a resolution of around 100 nanometer, so much less than just a single cell. So we can look at immune cell populations. And some examples of data that we've generated is CD3 polarization. So we've stimulated cells, treated them so we can actually create a cap on a particular cell. We've also done that with rituximab. That's a CD20 polarization event. So it's a drug that's used to actually target cells for directed cell death. All of these assays we've done in multiplex, and you can see a list of some of the antibodies on the bottom. If you need an exhaustive list, come by our booth or check our preprint. And you can actually look at the data in heat map view or these violin plots that show you each and every individual single cell. So you have in these events here, let's see, there, you can see the polarization event. You can also see that there's polarization in the heat map. And down here, you actually have a microscope image showing the same view. We can also look at chemotactic cells, uropod formation, treat the cells, and you can see where these proteins will aggregate and these proteins will segregate. So red is aggregation, blue is segregation. So they aren't up or down regulated. They move around on the cell surface, and then we take a snapshot and see how they look. And you can break it down into a component view or look at it in a forced spherical view or the heat map or even a network view of the same proteins. We can also look at some level of cell-to-cell interaction. And what you have here are two different cell types where you can see that they are interacting with each other. And we have a group of markers that make it so it stays on one cell or another, and we can actually separate them at the component view. Pharma is a pretty good market for us. A lot of people really like this because you can look at a couple of different compounds. As long as you design your experiment well, you can say these two markers were what they were looking at before on flow cytometry, but they weren't able to see the amazing bit of difference that they see with our 80 antibody panel. So we looked at 80 different markers in one assay at the same time on 1,000 cells in parallel. But we could also see things like differential polarization, which you can't detect in flow cytometry. And this compound 2 didn't show anything really, but our compound 1 did. And then also with the co-localization, we also saw a tremendous amount of segregation occurring as well as aggregation of proteins on the cell surface, especially with compound 1. So this particular pharmaceutical decided to go with compound 1, whereas in the beginning they were thinking about going with compound 2 because that's what they could see with flow cytometry. What is our product? What do we sell? We sell reagents. All of this is done without an instrument, so you don't need anything that's complicated. So you basically need some pipetters and a centrifuge and someone who is willing to run a sequencer for you. But this is our product. It's available now. It's 1,000 cells, 80-plex antibody panel. So if you have more questions, feel free to stop by at our booth, 612, and we'll tell you all about it. Thank you very much.
Thank you, and thank you for the sponsorship of ABRF. So we'll start with our first speaker, who's going to be Josh Stapleton from Penn State. If you have the app, his bio and interests, both professional and personal, are in there, so I'm not going to go through that. What I'd like to do, though, is just say a couple of things about the session. For those of you who were here last year, one of my goals has been to try and weave in materials scientists or non-bio people into electron microscopy at ABRF. I think there's a great place for them. So there is a theme running through the talks today, which is that in different ways, from training to research to instrument use of how bio and material science people can work together. And I think Josh's approach is going to be to talk to us about how Penn State is, and will be thinking about training in a cross-disciplinary way between the materials and the biologists. So Josh, take it away.
Okay, good morning, everybody. The lights are super bright, so if I put my sun, no, I'm not going to wear those. Thanks for having me here. This is actually year 2. I've been at ABRF and have really appreciated the opportunity to sort of mingle with folks very much like me, but very different than myself. Chris was very successful in identifying someone that knows nothing about bio, so all bio questions are completely off the table for me. I understood the word cell in that last talk, and then after that I was just thinking about some material stuff. So as he said, I want to kind of give some perspective on kind of interdisciplinary resources, interdisciplinary research, and looking forward to interdisciplinary training at Penn State. I'm with what's called the Materials Characterization Lab at Penn State. We're the largest basically shared research support facility on campus, but I don't want the name to mislead you. It's just sort of a legacy name, Materials. We've got a lot of materials activity, but we are very interested in supporting all researchers that come to us. Going to give a little bit of history and then kind of close out with some ideas that are very, very fresh and we're working on right now related to training and moving forward in the future. Figuring out how to change the slides. There we go. A couple slides to just kind of set the table for sort of the ecosystem that I function within. So I'm very grateful that I don't live within a single department, within a single college. Penn State has a 2-decade-plus legacy of an interdisciplinary institute system. Another way to sort of refer to these institutes as they're sort of the silo busters. So they have resources, they have influence, they exist to figure out strategies to get super smart adults in colleges and departments to collaborate, to work together, to share, to educate students, solve big problems. So that's a key part of how we function at the university. Specifically, I'm with the Materials Research Institute, but some of my most frequent sort of collaborators are in our Institutes for Energy and Environment, our Life Sciences Institute, and increasingly our Institute for Computational and Data Sciences. MCL is obviously not the only game in town at Penn State when it comes to core facilities. We've got a pretty broad reach of facilities at Penn State that range from nanofabrication to cryo-EM to microplastic specific analyses to MCL. I don't know why that's not, there we go. MRI is actually the oldest interdisciplinary institute at the university, about a 50-year history of enabling interdisciplinary activity. And it's really that foundation that we kind of work from at the university to try to bring folks together. At the moment, there's about 320-ish materials-related faculty at Penn State. We don't have a material science and engineering department with 320 faculty. That's sort of identified based upon research interests, and that is very broad. That's everything from people in food sciences to civil engineering to materials science to physics. Materials is fun because it is such an interdisciplinary activity. This is the laundry list slide. Also, I realize this is an electron microscopy session, but we are a lot more than electron microscopy at Penn State. We're really sort of the core facility for all things X-ray scattering, molecular spectroscopy, surface analysis, and electron sort of ion microscopy capabilities across the board. Really fortunate to have ongoing investment from the university to kind of refresh capital, add new capital, and at the moment, about 23 full-time staff within the facility that I'm a part of, engaged in a really broad cross-section of research. Like many other core facilities around the country, you know, we're a smorgasbord of chemists, physicists, geologists, biomedical engineers that are spending, you know, a decent fraction of our time every day training students how to apply techniques, but also serving as sort of independent investigators along with other researchers at the university. You've got a PI that wants to bring in AFMIR, so nanoscale molecular spectroscopy, and they're kind of looking for a collaborator. You know, we've got people at that level that get involved in proposal writing, get involved in helping to execute research on programs. So it's a very similar job. And I was reminded of this yesterday listening to one of the sessions about how similar everything really is, and the disciplinary boundaries I view as really more opportunities, not actually boundaries any longer. So a little bit of history, pre-2011, we had electron microscopes everywhere at Penn State. Even had the brilliant idea that at some point there was an innovation building that was built 3 miles off of campus because that seemed like a good idea. Very hard to get to. We had our nanofabrication facility out there. We had multiple electron microscopes. And so this was really the lay of the land of microscopy at Penn State. Then in 2011, we moved into what we now call the Millennium Science Complex. So the materials characterization lab, little blueprint there on the right, we're underneath that little dangling arch that you see on the bottom left image there is where all of the core facilities are. And you know, like a lot of you know, building openings at universities, there's a lot of grandiose verbiage and ideas about how this building is going to change the world and solve everything and, you know, cost a lot of money. And the vision was awesome, but it's one thing to spend a large amount of money and open a building. It's a whole nother thing to figure out how do you actually get the inhabitants of this building as well as the regional inhabitants on campus to actually work with each other. So spent a lot of time, as you'll see, spent about a decade, if not more, really trying to figure out, before we even get into overlapping training opportunities and sharing instrumentation, just establishing trust with people and being like, hey, we're actually on the same team. Like, we all work at Penn State. We're trying to educate students and, you know, push research forward. And the university is indicating a desire to invest in resources. We should stop fighting and squabbling and, you know, getting our money and running into this corner. Also, a little plug for ChatGPT. It's amazing the custom graphics you can fire up now using that. I said, give me 4 children crying and fighting over toys, because I felt like that can be a little bit of a good proxy for academia. 10 seconds later, my Microsoft Copilot spits that out. But it's been a lot of fun because where there's sort of these challenges and barriers, we've really kind of leveraged them as opportunities. And so going back kind of right after the building was built, one of the first things, the battery's dead. Really one of the first things that I had to do as sort of the flag bearer for the materials core facility was make sure I got to know the life sciences folks. So moved into a building, bunch of instruments there, but no real culture of sharing there. So myself and the facility director at the time, actually this is a long time ago, it's kind of ironic because it's kind of sort of a little bit of the title of this session and what I did last year, back in like 2011-12, you know, talking about we should share and there's opportunities. So really did start at the base kind of relationship building level, showing how there's a lot of mutual wins when these different disciplines come together. Very excited actually for a session that's right after this, I think. It's about crossover of materials and life sciences. We're doing a lot of that, which is exciting. And then kind of once the grassroots, you know, core facility people are commiserating and not arguing and fighting and collaborating, we really kind of just started to push ideas up and said, we think there's much better ways for organizing, resourcing core facilities at the university. So we spent a couple of years, this is back in the era of 4x3 aspect ratio PowerPoints. So you know, it's got a little age on it. my really amazing PowerPoint graphic design work you can see here, but it was really trying to communicate we overlap. We can do things together. There's a lot of science problems at the intersection of life sciences and materials where tools that might traditionally be in one domain over another could be leveraged by a broader community. So we kind of started this internal campaign in 2015, pushed on it a little bit more, came up with a catchy name because we felt like maybe that would be easier to sell it as we pushed it up. Common vision, pulled a variety of things out of the literature to show that folks are coming together. There's a lot of opportunity in science as folks are coming together. And again, pushing ideas up, and then finally, really got some traction amongst multiple institutes. So this is where the institutes are also really valuable for us at the university. I didn't have to go around to convince a bunch of research deans to put some money on the table. We were able to do this within the interdisciplinary institute system. And so we pulled together an initiative that was a whole bunch of, not a whole bunch, one or two symposia that were kind of highlighting via lightning talks and posters, introducing techniques to disciplines that don't really use a technique, and really kind of started to fund a lot of interesting interactions, which was very exciting to sort of finally see, people crossing over the aisle, if you will, leveraging tools and approaches. And so a little bit of a, intro of a joke there, but, a microbiologist, surface scientist, and a chemist, walk into a bar. And what do? Well, they write an NIH S10 proposal. And this directly came out of, a couple of years of pulling people together, getting some seed grants on the table. And I was 100% convinced that this proposal was a complete waste of time because the first sentence in a proposal to NIH said, the materials characterization lab. And I was like, there's no way they're going to fund this. They're going to be like, why would we give money to materials people? However, they did. And so super exciting to kind of see, and this is one small win, sort of building this culture and making the case to a federal agency that even though we don't know bio, we have the core expertise in sharing the instrumentation well. That panel agreed with us, awarded that, and now sort of 6 years later, what's really interesting, this is a snapshot from a couple of the, from our AFM group within MCL. And they're doing all kinds of stuff. I mean, we've since bought AFMs with confocal optical microscopes because the biologists want to sort of do, mesoscale localization before they go in and do high-resolution AFM. And more importantly, it really actually helped us justify hiring a very different skill set into the materials characterization lab. So Sarah Kimley is a PhD biologist. And if you had read the job posting about 5 years ago, we were very explicitly looking for somebody that wanted to work at the boundary and could help translate biology problems, to the rest of the facility so we kind of understand what are the problems that folks want to work on. And then she kind of translates the other way. So I felt like we hired a translator. People has been super key with some of our success in this space. So a little bit on electron microscopy. This is sort of a rundown, again, sort of 10 years, 11 years after we started this. It was nice to see in a Microscopy Today article a couple of years ago, it doesn't seem like much, but in the figure, hey, look, we're sharing instrumentation where you never would have seen this figure 15 years ago. So this felt like tiny success, and we're adding some additional microscopes down there. It's getting a bit full. And then to sort of kind of emphasize the importance of people in all of this, this is a current kind of rundown of SEM dual beam fibs in the building. And what was really, what's been one of the opportunities of all of the, what I'll call personnel churn of the past couple of years is that we were really able to think about staffing in some different ways. And so the two circles on both sides of the slide are actually the same broader organization, the Materials Research Institute, but we were very poor at sharing people across the boundaries. And so it wasn't until about a year ago that we said, the nanofab has engineers, that are working on processing, that are working on metrology. MCL's got people that are working on characterization. Why don't we actually share these people? And it is, it's a little funny, a little disappointing, but also feels like, wow, there is some serious low-hanging fruit out there. So about a year ago, we brought in one of our engineers from the nanofab, whose primary responsibility before was just microscopy and thin film deposition in our nanofab. And now he's split into MCL, where he's running SEM and FIB. And the hilarious part of this is there's now all these people in our nanofab that are like, where did this technique FIB come from? How long has Penn State had FIB? Because it was sort of handed to them on a platter, and it's like, look, this is how you can use FIB to look at defects in your devices. We've had a FIB at Penn State since 2005. Bangji Liu, who is this person helping to cross over the boundaries, he's got a slide deck of like 30 examples now, just in the past 9 months, of like the benefit of being in proximity to people that are working on a project and handing them a solution. And so it's kind of been exciting. Again, a little bit frustrating that it's like, really, we couldn't, and our nanofab is 1 floor above characterization, so it's not even like we're in separate buildings. So a lot of kind of optimism when you bring the right people to the game. A lot of great sort of science to still be pushed. All right, switching gears a little bit to training, education. We have this, you know, kind of grandiose but sort of genuine sort of mission statement there as to like, why do we do what we do in MCL? And really, it is all about educating the next generation to solve problems. However, it's not always that easy. You know, you've got these actors out there called time, money, proposals, impatience, pressure, publish or perish. And so we're not always you know, able to invest the amount of time that we want as a core facility and every user that comes through. And so we're sort of asking ourselves, are we just generating safe operators, meaning they know which buttons to turn, they're not hurting themselves, they're not hurting the instrument, or are they skilled practitioners? And the answer is like, well, it's kind of somewhere in between, and it really depends. So for us, we're right at the beginning of kind of an entire revolution of training and education within the facility and how we're going to approach it. And this is not an exhaustive list of the challenges. It's just kind of a couple lists of the challenges. And that is, at Penn State, we're not short on classes that introduce fundamentals of techniques. But MCL really is the place where folks want practical knowledge. They want the tips, the tricks, the arts, the crafts. Like, how do you actually do this? The facilities are really the place to do that. So how can we do that at scale and at a price point that, you know, the faculty can afford, and that we have the capacity to actually deliver that? So, you know, our cost model is challenging to education. I get it. You got a brand new student, and all of a sudden they're paying $50 an hour for an instrument, $50 an hour for staff time. $3,000 later, you might be saying, I really want my student should be trained by now. And it's like, well, we can only meet the student where they are and try and lift them up. And so part of a couple of the components of sort of our strategy moving forward are, we're really looking at acquisition of training-centric instrumentation. So we're looking at cost of ownership. We're looking at, we need to give users a lot more stick time on various instruments. We want to design content that is not 100% staff delivered. What's the right balance between really good sort of Canvas modules and students with you know, hands-on time in the instrument? And then looking at kind of meeting people where they really are in terms of how much extra training they can afford. And that could even just be in the sense of time. Like, even if we offered 25 hours of free SEM training, I know that not every PI at Penn State would say like, oh sure, your student can take 25 hours of training. So how can we develop some modular content? And then we got to find resources to enable and fund all of this. So in my world, I realize this is a community that probably couldn't care less about silicon carbide. I'm not a, you know, we're chasing resources to some extent. But what's been really interesting, and I heard a little bit of this yesterday about core facilities becoming flexible and thinking about new opportunities. Workforce development, my joke on campus is it's the new nano. It's like every proposal that's going out now is workforce development, workforce development. The amazing news, in my opinion, for core facilities people is I think we've got a lot of solutions that people are looking for in terms of training up the next generation of researchers. So I'm sharing this just because it's one pot of resources that we're pursuing to enable essentially a new structure at Penn State. So this is, I think, my last slide, but in kind of simple terms, what we're looking at is, at the present, we train users to become independent operators. That's the blue box at the top. Let's just say it's SEM, 4 hours, you're an independent operator on the instrument. However, we know that you're just an operator. You know, there's not enough time for you to run through our reference samples, to learn tips and tricks. And so we're spending time kind of developing what we're calling a content buffet. And that is, you know, up to maybe 20 hours of content that's sort of fundamentals as well as applications. And then we're going to create some ways to deliver that content kind of based upon where people are. So we've got certain students that are still seeking credits, you know, so early in their career, graduate students, they might say, I need to fulfill 3 additional credits in MAT-C. I want to take an EM class, an IR class, and an XRD class. We're going to get involved in delivering that content. There's other current users of the facility that say, hey, I need to just get trained on an SEM. I'm not going to go through 20 hours of content, but we want to do better than the 4 hours right now. So they're going to have a smaller portion of the content buffet where we're going to, I don't want to say force, but we're going to provide an opportunity for the real understanding of tips, tricks, and how you do these things. And then also looking at sort of a third option as we're talking to industry, as they're hiring people into some of their corporate analytical functions, they're saying like, man, we're looking for like a boot camp. Like if we could send our new hires to a place that could help them practically understand how do you use XPS and FTIR to solve problems, we would pay you out of our, you know, professional development budget for that type of training. So nothing overly grandiose here, just very different than a standard core facility model, which is really the green sphere is how we functioned in the past. We're really looking to expand the ways that we're delivering this content across campus. So with that, I promised to finish early. I got 21 seconds, so I'm going to wrap it up. Looking forward to a panel discussion later today and hopefully meeting some new faces out there, so thank you.
Great, thanks Josh, that was right on the mark, very good. Our next speaker is Kirk Czymmek, and by way of introduction, I'd like to introduce the concept now of volume electron microscopy. This was identified by Nature last year, 2023, as one of the 7 techniques to watch. And for those of us who've been in electron microscopy for a while, there is, I guess, a long history of 2-dimensional imaging. And so that the whole concept of volume EM is exactly as it says, to look at volumes with lots of Z as well as X and Y. There are a whole range of different ways of approaching that. And again, Kirk's going to go through and talk about some of the ways that can be used. So Kirk, tell us about volume EM.
Okay, good morning, everyone. Thank you, Chris, for the introduction, and ABRF for having me here. I actually wanted to start off with a demographic question. Could you raise your hand if you're currently or recently doing electron microscopy? All right, so maybe 30%, not bad. Okay, and how many of you doing volume electron microscopy? Not too shabby. Okay, cool. For the rest of you, though, you'll learn something, and maybe for those folks who are already using volume electron microscopy, you'll see something that, interesting or new. So I'm going to start mostly facing this way. Apologize for that, folks, if I use my laser pointer, and I'm hitting the forward. There we go. Okay, so the first thing is what is the definition of volume electron microscopy? And to be honest with you, if you've been doing serial section transmission electron microscopy over the years, slow, tedious process typically. But if you captured volumes that are typically greater than a micron up to even to the millimeter scale at nanoscale resolutions, you would classify as volume electron microscopy. So that's not to be excluding the electron tomography, but that wouldn't classify because generally you're working with much thinner slices, okay? The second thing is, why would we use volume electron microscopy? And well, first of all, I think everybody appreciates life is inherently 3D. But the other thing is, I love this example of a cube in which if you sliced it in 2 dimensions, depending on how you hit that cube, you could get very different representations of what that object actually is, okay? And in the world of biology where people probably aren't taking enough statistics and trying to do mental gymnastics to get back at that. I just wanted to give you that to appreciate what's the value here. The other thing is, since there's about 60% of you who are not electron microscopists in here, and I wanted to actually show this more traditional sense, and I'm going to get the laser pointer. So you have a normal TEM where you're cutting the sample into a very, very thin slice, typically under 100 nanometers. You put heavy metals in it, and you pass an electron beam, and you can get an image on a detector or camera. Traditionally, for scanning electron microscopy, instead of passing the beam through the sample, you'll actually focus it to a small point and scan it at a raster scan. So a lot of folks who think about SEM and life sciences are thinking of these surface types of images. But it turns out that if you stain the sample with heavy metals, and then you section off the surface and you look at the block face, you can use backscatter imaging mode to get a very TEM-like image. Won't be the same resolution, but it'll be pretty close. So again, the electron beam comes down, secondary electrons which are coming near the surface will give you this kind of image, and since I'm a plant biologist, you'll see some of these images sprinkled, but use your favorite cell here. But if you have heavy metals, and the beam is coming down from deeper down into the sample, you'll get backscatter electrons, which are going to come back at the same energy in which you're putting into the system. So you can differentiate those and get a really cool image like this. Well, now that I can get a TEM image like that, so what can I do next? So this is where the technique of volume electron microscopy comes in. And I will tell you that there are volume electron microscopy TEM techniques, but today I'm going to focus on the scanning electron microscopy versions. Because they're, I think, more universally possible and easier to implement. So I will be talking about a technique called array tomography, focused ion beam scanning electron microscopy, serial block face scanning electron microscopy, and then if you take a focused ion beam SEM and you put it in cryogenic temperatures, you can get cryo-FIB-SEM, okay? Now, as already mentioned, Volume EM last year got the 7 techniques to watch. In 2023 with CRISPR and the James Watt telescope. That's pretty good company, folks, so stay tuned. Hopefully we can have just as equal discoveries over the coming years. So I'm going to start with array tomography scanning electron microscopy as the first step. And basically, oops, I hit the wrong button, but that's okay, I'm going to go with it. Basically, the electron beam comes down, and your sample is on a hard surface. It could be a cover slip, it could be a semiconductor, and you have long ribbons of your sections. And if you look on here, you can see all of this multi-scale imaging. So the long ribbons, this is actually one of my plant samples, looking at a root nodule. So those who like legumes to eat their vegetables, this is what's pulling in nitrogen from the soil and giving it in a usable form for the plant. But essentially, you have these long serial sections onto a surface, okay? So let's go to the next slide, and I just have to push the right button. Now, so basically, you collect a ribbon of serial sections and onto a substrate, and you can probe it a couple ways. For those who are familiar with multiplexing, you can use fluorescence probes, strip it, reprobe it, strip it, or you can stain it with heavy metal stains and look at it like you would normally use a TEM image. Or you can get really fancy and you can do all of this fluorescence multiplexing, and then you can go to the EM and look at the affiliated structure. A little bit fancy. On here I put a few papers on here. Occasionally that would be a useful resource for you. All right, so I'll go to the next slide. So by the way, one of the things that it's important for this to happen is if you have a laboratory in your facility that has an ultramicrotome, you can see how we can collect all these nice ribbons into long rows. In this particular case, they're collecting on a semiconductor wafer. Now, the cool thing about this is it's archival, which means you can take those, you can interrogate them, and you can come back later on in case there's some new insights, okay? So when you're done, you get these sections, and of course, anybody who's sectioned before knows that there's things that happen during sectioning. There's wrinkles and ribbons and tears. And you have to align them. So here you want to acquire the data. In this particular case, it's representing a block of about 30 microns cubed. You have to align that data, and there's software tools out to do that. You then need to segment it and define what it is that you want to pull out of that volume. You can quantify it and then visualize it. And I like this nice next little cartoon here. Here's an example of a brain slice where they're actually trying to do connectomics. And I have to be honest with you, a lot of volume EM was driven by the National Institutes of Health going after the connectome because, you know, trying to look at synapses is a nanoscopic type of adventure, and you can use this type of tool for it, okay? So that's array tomography, ribbons of sections, okay? So the next part of it is serial block phase scanning electron microscopy, okay? Now, in this particular technique, what you do is you have the electron beam just like before. You will have your sample on a little post, but you actually have an in situ microtome, ultramicrotome. So rather than the technician sitting there over a boat of water trying to collect sections and ribbons, you actually have a diamond knife inside that's shaving off the inside, the section, one section at a time, and I have a little cartoon of what that looks like. So here's the sample sitting on a pin, and it's very opaque on here. Here's the diamond knife. It just shaves across the top of the sample. And then the electron beam comes down and raster scans back and forth, and then you can create each slice. Now, the cool thing about serial block face image is the block face is rigid. So it's much easier to do alignment. You're not going to have some of the distortions you would have from array tomography. The thing that's a little bit different about it is the sample's gone. Once that section's gone, it's actually in the column somewhere. So you can't archive it per se. But there's a lot of advantages, and it's very fast. In an overnight run, I can typically get, 500 sections, and trying to do that on a TEM, it would really be weeks worth of work. So one of the things about these techniques is that with array tomography, you can usually use typical TEM type protocols to stain them and get contrast. With serial block face, although maybe not have been obvious from my picture, the sample is quite a bit away from the detector. And that's to get clearance to put a diamond in there sectioning on the material. And because of that long distance, you need more stain in the sample. So I would tell you that one of the things that have moved serial block face along is the fact that we got more heavy metals and protocols that will allow us to enhance those heavy metal stains, okay? I'm not going to get into all the details today, but just to give you a sense that this is our toolkit we use to get strong signals. And if you've done TEM before, we typically take a living cell, we fix it with aldehydes for quick fixation. We would then use a heavy metal called osmium tetroxide. Gives you normal, really great staining for TEM, but we also go in and amplify it with a mordant called thiocarbohydrazide, so we can actually stack osmium on top of each other. We can also put in other heavy metals like uranium and lead, and when we're done, we have pretty much a very opaque sample. And we put it into resin. So again, we have to put these extra steps. A normal TEM block is a little bit trickier to get good signal to noise. All right, I won't get too much more detail and technical, but here's a journey in a mouse brain. Again, here's a neuron example. You can see all of the slices through this particular large volume. And they were looking at the connections in the neurons, but at the same time, they wanted to also look at mitochondrial density, and this is just the incredible amounts of information that you can get through the data, and of course make these beautiful 3D fly-throughs, but that have actually biologically useful information for the scientists that are doing it, okay? I'm going to go a step further and say, when I was doing this in the late, I don't want to say 1900s now, the late 2010 area, I actually had a corral of students who were going through and doing all that segmentation by hand. Increasingly these days, we're starting to use AI where you can train a few dozen images. Sometimes there's even models out there that do a better job. Here are some cancer cells in which we wanted to look at the connection between the endoplasmic reticulum, which are all these little multicolored, and the mitochondria. And again, the resolution is sufficient in the volumes that we can actually find and count all those contact sites and things like this, just to kind of give you an example. Again, you may not looking at ER mitochondrial contact sites, but if you're trying to look at things that are close proximity, you can use this kind of a tool. Again, that data set was collected in an overnight run using a volume electron microscopy serial block face technique, okay? All right, so now I'm going to move into FibSem. I think I'm on good cadence here, so we'll get through it. So focused ion beam scanning electron microscopy. The reality is that when you use a diamond or physical cutting, you really can only get down so thin. It's a matter of physics. 50 nanometers, 20 if you got a really perfect sample, but more like 50 to a little bit higher. But the ion beam is cool because you can shave just a few nanometers at a time. The other advantage is that, of course, the resolution of a scanning electron microscope is down to about a nanometer or two, plus or minus. With an ion beam, you can now get isotropic voxels. So you can look at your structure, and they'll have the same dimension, X, Y, and Z. That's not quite possible when you're trying to do the highest resolution work with a diamond-based sectioning tool. So I'm going to show you a little cartoon really quick on here. So in this particular implementation, there's a platinum pad that's put on the material. Some markers are put in for fiducial, so you can kind of track your movement and so forth. And then the ion beam is like a windshield wiper. It's just ablating away material to kind of clear a line of sight for the electron microscope. And then you start actually taking image after image just by removing off and ablating these thin layers at a time. Okay, so there's the SEM image would be imaging, and then you would, again, alternate imaging and ablating. So let me go in and skip forward to this next slide. So here's a beautiful example of a HeLa cell at about 8 nanometer isotropic resolution. And now you get these full 3D volumes of your cells. Okay. Again, depending on what you're looking at and trying to quantify out of here, taking just one of those slices would be beautiful, but taking it all in 3 dimensions, you can now see the power of actually understanding your biology. So many other experiments you can open up. Okay. I actually did some work with the yeast. This is 3 nanometer isotropic. This was in around 2012. So I labeled all the organelles inside of here. The endoplasmic reticulum is in yellow. The nucleus is in blue. The mitochondria, a lot of ER contact sites. But the cool thing about it is that this technique is quantifiable. So once you segment all these structures, you can start measuring it. So if you have phenotype that you expect that there's some effect that you want to measure on here, you can use this technique and see any kind of measurement you can from this three-dimensional structures. And in the spirit of this cross-disciplinary idea, actually I've done some soft materials with chromatography particles. If you can contrast it in backscatter mode, you can section it, you can use a serial block face or array tomography technique. And I've done like chromatography materials looking at absorption and all the pore size. But in this particular case, if you're working with hard materials, the focused ion beam is an ideal tool because it won't break your $5,000 knife or whatever your knife you have. So use the ion beam here. They're using steel wells in this particular case, and they're looking for the distribution of the different elements that go into your well when you solder these things. And then here's another example for our geo folks on here. A lot of the folks in shales are going to pull samples from under the shale and look at the porosity to see how much gas might be there versus other types of natural resources we need for energy sector. All right, my last section is cryo-electron microscopy. And I just invested in one of these things. It's way cool. It has some distinct advantages that I'm going to talk about. Number one is and there's a cryo-ET, I think there's a number of folks in here, the Nobel Prize in 2017. Of course, with cryo, you have the idea of getting things that are lifelike. I am not a single particle analysis person. I am an in situ cryo person. And I want to remind you, you can make lamella and so forth on these tools, but you can also get volume data and you can put it both together. And here's an example of an algal sample in which we're imaging unstained, it's frozen hydrated at liquid nitrogen temperature, and all the structures that you're seeing, the nuclear envelope, the nuclear pores, the stacking of these membranes called grana, they're 10 nanometer spacing on here, so you can resolve very easily down to lower levels. This is in a paper that I just published with for plant microbe interactions, and now you can see and rebuild a full algal cell. And now start quantifying the biology. So we're going to do things like environmental stress, heat stress, and so forth on it and make real measurements. And I want to go a step further. There's an article out in Structure called Multiscale 3D Cryocorrelative Microscopy for Vitrified Cells. I highly recommend that you go to Abigail Jean Leighton's talk in Lakeshore A after this session. She'll talk a little bit more in detail, but the cool thing is that you can take cryofluorescence data, which is down here, take that same fluorescence data mapped over to 3D volume EM at a cryo mode, and then you can go to structural biology at the same place. And I think if I push the button, you can actually see there's your frozen cells vitrified on a surface. The ion beam comes in and mills. You image the surface. You keep watching because you can see contrast it, right? So you can see where you're going. You can get to your target, which may be a fluorescent protein. And then once you get your target area, then you come in and mill it from the other side so you can make your lamella. And then finally, you can take the sample and do your cryo-ET. Okay, pretty cool. And of course, our system has an integrated fluorescence, but you can couple fluorescence microscopes under cryo mode afterwards, and you can help target what you need to do when you go to the structural biology. Okay, so I think my next slide, okay, hopefully it didn't jump forward. Okay, if you are interested in volume EM, we received an award from Chan Zuckerberg to build the volume EM community. We're building a ton of resources because, listen, the challenge of getting to volume EM is there's a steep learning curve. Back when we started doing this, everybody was working in an island, but I would highly recommend that you come on. We have an emailing list, a Twitter account. We're actually building software like a sandbox where you can come in and try the tool that will help you manipulate or do AI on it. So stay tuned, get on our lists. We'd love to have you to join us and be part of our community group. And then the last thing is Wiley has a little session on, and this is biological mostly, but there's several more series that are coming in where you can get experts from around the world, and they're recording these videos if you want to get a little bit more detail if you found something that's interesting today. And I'm giving you back 1 minute and 50 seconds, okay? Thank you very much.
Great, thank you, Kirk, and I can certainly second joining the volume EM community. There's some great material there. I've signed up. You should be able to find Purdue on the map of volume EM around the world. So the more the community builds, I think the more that we're going to be able to get out of volume EM. And just from a personal perspective, I have a serial block phase system, but with the ultramicrotome, but my facility also deals quite heavily with the materials community, either soft or hard, and we have dual beam fib and the software for doing those volume EM things on materials as well. So it really is a technique for cross-discipline. Okay, so final talk today is going to be from Nick Seaton, who is a local. Local in that he works at the University of Minnesota, but you will, as soon as he speaks, hear that he's much more of a local for me. He's originally from the UK, which I actually didn't know when I invited him to speak here. But the final talk is going to be on how to use a particular, really innovative scanning electron microscope for materials research. So it's my way of trying to get the biologists here to think about how material scientists use exactly the same technology and techniques in their world as we do in bio. So Nick, take it away. Thank you.
Thank you, Chris, for the introduction. So yes, my name is Nick Seaton. I work at the University of Minnesota in our characterization facility, and approximately a year ago we finished installing our new Thermo Fisher Aprio 2S low vac SEM. Prior to that, all we had were high vacuum SEMs, in which of course you'd need to coat your materials. So we're going to talk briefly about how SEM works for anyone who's not done any SEM. Then we're going to go on through why would we use low vacuum SEM, and then we'll do some examples of projects that we've been working on. So obviously, we can see we have a diagram of an SEM on the left. Why do we use electrons? Well, because we got the smaller wavelength, and it allows you to get better resolution. In material science, people keep pushing to smaller and smaller scales, and we need smaller and smaller probes to actually visualize the materials that they're making. SEM is very useful in that we can raster the beam across a large area at very fine detail. We get the secondary electrons that give us our surface topography. We get backscattered electrons for compositional contrast. And then there are other signals such as X-rays for EDS. We can get diffraction for EBSD. And the benefit of SEM over TEM, as I always tell people, is the reasonably fast sample preparation. It can take sometimes up to a week to prepare a nice TEM sample if you're doing the old-fashioned mechanical polishing method. But in general, in the low-vac SEM, I can pretty much take any random sample someone brings me and just put it straight in there and image them. So here's just a quick example of what secondary and backscattered electrons are. The secondary electron is an orbital electron that's been ejected from the material. The backscattered electron is a beam electron that's been bent around the nucleus and comes out. There's an energy difference between them, and that's how we detect them. As a quick example there, secondary electron image on the left shows us nicely the topography. This is actually just the corner of a letter printed on a piece of paper with a standard inkjet printer. You can see all the lumps in the printing, you can see the fibers in the paper, but then in the backscattered image, the printed ink is much, much brighter, denoting that it's made of heavier atomic number elements, and so we get a compositional contrast image. And the combination of the two images really tells us an awful lot about the material. So, yes, why do we need low vacuum mode? Well, conventional SEMs, as I said, work in high vacuum, and when you're shooting electrons at a non-conductive sample in high vacuum, many of them get stuck on the surface of the material, and then as you keep trying to shoot more and more electrons at them, they build up, and eventually they distort the scan line of the image, and so we get image distortions. And typically this got around by putting a heavy metal coating on the material. Most people started using gold. People have moved to things like platinum and iridium and even osmium, trying to get finer and finer grain sizes, because what you don't want is for your coating to have some feature that you can image that you then mistake as part of your material. Low vacuum SEM was invented a surprisingly long time ago, back roughly when I was born in the late 70s to early 80s. But a lot of people didn't use them at first. They were considered sort of a biological thing. Oh, you can put your bio stuff in there, and the material scientists kind of ignored them for a while. And I say, we only got our first one a year ago here. So the low vacuum SEM works by essentially bleeding a gas into the chamber. In the case of our SEM, we actually use water vapor. It's very easy. You just have a little Erlenmeyer flask of water connected by a needle valve to the chamber. The computer controls the needle valve, opens and closes it to let the water evaporate into the chamber. This has two effects. One, as the primary electron beam enters the chamber, it scatters off some of the gas molecules, ionizes them, and those positive ions can then recombine with the electrons stuck to the surface and carry the charge away. But also, you get a boost of the signal. As the secondary electrons exit the material, they can hit other gas molecules, generate more electrons, and you get a cascade amplification of the signal. So this is a picture of our SEM, and a little diagram on the right shows the positive ions being created by the beam, recombining with the sample's charge electrons on the surface, and the cascade amplification of the outgoing signals. So standard SEM on the left, you've got various pumps at the bottom, usually these days a scroll pump and a turbo pump to keep a nice clean chamber, no oil involved. Your samples would sit somewhere about 10 to the minus 4 Pascal range, and then your electron column is typically pumped to higher vacuum, 10 to the minus 6, 10 to the minus 8 for field emission guns at the very top end. With a low vacuum SEM on the right, there's a couple of extra chambers at the bottom of the column. These are differential pumping chambers. They enable you to maintain the ultra-high vacuum in the column so that the electron gun keeps working whilst maintaining a relatively high pressure in the sample chamber. In the case of our SEM, anywhere from 10 to the minus 4 up to 500 pascals of water vapor. This is the view of the actual vacuum diagram from the microscope. There's an awful lot of valves and pumps and things in there, but it's all completely controlled by the computer. On the left, you simply select whether you want to be in high or low vac, and you pick the pressure you want to work at. You can just type a number in and press go. It takes about 30 seconds to change vacuum modes. So obviously, the very first things we did in low vacuum was actually biology. Someone from the entomology department here was very interested in sensory papillae on the antennae of box elder bugs, and they're comparing males to females and the ventral or lateral sides. And so we put whole heads of box elder bugs in there and spin them around and look at them to try and find the tiny little sensory papillae in between the hairs on the antennae. I also had someone from the bio department looking at artificial meat from plant proteins. They actually brought them to me native state. They were little sort of 5mm cubes of wet, squishy stuff. They went in the SEM, we imaged them. They did admittedly come out dry and crispy, but they were native state when they went in. More of the work I do is in the materials side, though, and so here we got bismuth oxide on silica particles. This is a catalyst being investigated, and depending on the beam conditions, you get a very different view. So on the left, we have a picture that is at low beam power, low beam current, and we get a lot of secondary electrons. You see a lot of topography, but we don't get very much chemistry. As we turn the beam power up to higher currents, we get proportionally more backscattered electrons on the right-hand image, and the brighter particles are actually the bismuth oxide on top of the silica. So this was very helpful for them to see the distribution of their catalyst on the silica substrate. This was from a local company. They were doing laser engraving on a polymer. This was actually particularly hard because you can see these squares with the naked eye, and they're almost invisible in the SEM. They're exceptionally shallow. I think the topography that's being viewed there was approximately 5 nanometers or less. In the end, we ended up using AFM to actually get a better view of what was going on, but they had all these squares lined up on this transparent polymer, and we were kind of going along and seeing if we could see the marks created by their laser, and looking at the damage of the surface of the polymer. You can see in the zoomed in view on the right, there's a lot of cracks developed from the laser as it heats up the polymer. Working with another local company, these are oxygen sensors from ventilators that they started making rapidly during the pandemic, and they were having a trouble with some of their sensors failing. So this is actually checking the wire bond for the sensing wire at the top of the ceramic and polymer posts, and we can see that there's an awful lot of material peeling off around the edge. So there's a good reason for why this sensor is failing. There's no conductive path for the sensor there. EDS, energy dispersive X-ray spectroscopy. This is a very common technique in material science in SEM. When the secondary electron is generated, you've left a hole in the inner orbital of an atom, and an outer orbital will drop in to fill that. To do so, it must release a photon of energy. In core shell electron transitions, these photons happen to be X-ray wavelength. With a detector, we can pick up the X-rays, we can measure their energy, and therefore we know which atom they came from. If you combine this with the image of scanning the beam, you can generate, not going to go forward, there we go. You can generate pictures and spectra of the chemical distribution in your material. The actual detectors themselves are relatively simple. There's a silicon lithium detector. It's kept cold, typically these days just by a Peltier cooler, but in the past with liquid nitrogen. And they simply point at the sample and they just collect the X-rays that hit the detector. And the computer combines that with the position of the beam to give you where the chemistry is coming from. They can collect in many ways. Standard analyzer mode is just everything in the field of view, scan the beam over it, give you a bulk composition. We can go to point and shoot mode, where we can put the beam down in an individual spot and point at a piece on a picture and say, what's that? Line scan, we can do a bunch of points in a row and generate a chemical profile. And mapping, we can scan across the whole area and give you 2D spatial distribution of elements. So this is an example here. This was a piece of polymer that is put into a water well with a bunch of glass beads to simulate sand, and they spin it around for a few months. And when we take them out at the end, we find a layer of silica or glass on the surface of the polymer. They were expecting that the glass would wear the polymer away, and what's actually happening is the beads are wearing each other away and depositing on the surface of the polymer. We did this with just regular SEM, but they wanted to know, well, how thick is this layer? So I put it into my fib, I put down a protective cap of platinum, sliced it open, but my fib doesn't have EDS. So I just took some pictures in backscatter, and we can see the bright layer beneath the platinum cap there above the darker platinum, which I thought pretty much proved it was glass, but they insisted I take it and put it in the other SEM and do the EDS on it. And you can actually see these little parts there in the silicon map show where the glass is. Those 2 bits there line up with the 2 bits in the backscatter image, therefore proving that it really was glass on top of the polymer. EBSD, electron backscatter diffraction. This is the technique I specialize in. It's used quite extensively in material science and geology. But essentially, we shoot the electrons in at a very shallow angle, and we can collect a diffraction pattern out. And the diffraction pattern is actually the direct projection of the crystallographic planes in the material. If we take a model and we spin the unit cell around, we can see how the pattern would look, so we can model what every angle should look like. And therefore, when we point the beam at a crystal, we can collect the diffraction pattern, and the computer can then recreate which orientation that crystal is. When I started doing EBSD, it used to run at about 3 points a second. The latest detector I have now runs at over 5,000 points a second. So this is a map from the mineral olivine. It's a magnesium aluminum silicate. And the colors here represent the different orientations of the grains. So this whole map was collected in about 5 minutes at pretty high resolution. We've got a 100 micron scale bar at the bottom. I believe the pixel size is probably about 1 micron. You can't get quite as low a resolution as you could with regular SEM imaging, because this signal has slightly worse resolution due to the high tilt. The beam spreads out a bit in the y-axis, and also because it's coming from a deeper part of the activation volume. So with more material in there, it's just a bit worse resolution. But you can generally get between 50 and 100 nanometer resolution at best. This is an example from geology. This is the mineral apatite, which biologists may know as your bones and teeth, but in geology, we also find it lying around in rocks. And this was a piece we did a deformation experiment on. So we took a piece of apatite, we pressurized it, we heated it, and we twisted it at the same time at hundreds of megapascals of pressure. The very left-hand image is one side of the crystal, and we see it's all one color, representing that it's a single crystal. On the other side of the sample, we see there are different colors. And this tells us that there were 2 different crystal orientations. And between them, we can see a small band of multiple colors. If we look at the larger zoomed in image, we can see all these little colored grains that have appeared. These are actually recrystallizing grains during deformation of the material. So this is a snapshot of recrystallization happening. This is recent work I did on a meteorite NWA5315. These are known as clouded pyroxenes. The top maps, the red and blue ones, are phase maps. The red is a mineral augite, the blue is the fabulous mineral pigeonite, and the green is chromite, and the yellow is ilmenite. And this is what we were actually looking for, were these little inclusion grains, and trying to see where they were and what orientation relationship they had to the other grains. We can see at the bottom we have orientation maps, so we can actually take this data and plot pole figures and actually look at the orientations in much more detail, but we can also just look at the pictures. The other interesting thing we found in this sample here, normally the red and blue minerals will separate from each other in these nice straight lines. They're crystallographically controlled. Unfortunately, in this area, we found some curved ones. We have no explanation for what those are yet, but we're working on a paper for that. Not quite sure how you can make a curved crystallographic plane when the others are straight. And that's it. I will point out I will not be here for the discussion session this afternoon, so if anyone has any questions, I can either answer them now or you can talk to me outside, but I have to catch a plane shortly.
So a little contribution from me now. In case you haven't joined the dots yet, if we think about those last two talks, Kirk talked about finding structure and composition in volumes. What Nick showed was primarily a single surface and all of the different chemistries and crystallographic structure that can be obtained. Let's put all of that together, because if we can do all of that in a single plane and then use volume EM, we can now get volume of chemical composition and crystallographic structure. Kirk also talked about the need for additional signal by putting heavy metals into samples to get good imaging, but that also has another factor for volume EM, which is you don't need to do the typical sputter coating for that we would do for non-conductive SEM. But we've just heard from Nick's stuff that you don't need to have that metal coating if you're using low vacuum SEM. So now we can put all of that together. We can do non-conducting volume EM by using low vacuum. There are some limitations with resolution, but we can then add in all of this additional spectroscopic data, crystallographic data, and make 3D maps. Nick just showed some meteorite examples. Last year, I had a moon dust particle collected by Apollo 17, and we did 3D maps of a single. NASA were very reluctant to let it go, particularly as we weren't going to give it back because it was destructive, but we did 3D chemical composition through volume EM of a moon dust particle. So think about adding those second two talks together and think about the total information that we can get from samples. 17 seconds over. Good timing for a session. So as Nick said, he's got to disappear, catch a plane later. If you have questions for him, I would encourage you, if you have more questions, to come to our breakout session this afternoon, which is somewhere at some time, I forget. 2 o'clock? In right. So questions later or over lunch, or and thank you for attending. And thank you to the speakers, and as always, our sponsors that make the whole program possible.